really excited to be here with you guys today. I'm going to be talking a little bit about my pathway into science, some of the things that kind of motivated me into science. And then towards the middle, I'm going to start talking about CRISPR and the potential of CRISPR to really change and revolutionize the world and how it's already doing it. So I was born and raised here in Costa Rica in the middle of the Americas, a small country with about 5.1 million people. It's a country that's where we're really connected to nature. And I think this connection to nature really fosters curiosity in a very natural way. It was really great to grow up here with the beach, with volcanoes, rivers, and it's also the land of pura vida. Pura vida is means pure life, and it's really a way of living, a way of thinking, a way of kind of enjoying your day to day very well. And this is where it all started. And I had the fortune of growing up in Costa Rica in the 90s when there was this really famous Costa Rican, Dr. Franklin Diaz. He was born and raised in Costa Rica, and he's the first astronaut that was born in Latin America to go to space. Since then, he's gone to space about 7 times. I think for a long time, he had the record of going to space the most. And he really, for me, was an inspiration because sometimes when you grow up in small countries or communities where you're not directly connected with people who are scientists or people who are changing the world, you may think that those things are not achievable. And he, by going up to space, showed me that if this person who grew up in Costa Rica was able to do it, then I was able to really accomplish anything I wanted. And then I first started getting interested in genes when Dolly, the first mammal, was cloned in 1996. That's already a long time ago, but at that time I was only 8 years old. I didn't know what DNA was. I didn't understand what cloning was, but I knew that this was something big. It was really exciting. And it really stuck with me that moment when this happened. And it really kind of sparked an interest in genes and genetic engineering. And then the other thing that really, I think, was important in the 90s was HIV and the search for a vaccine and a cure. During the 90s, the epidemic of HIV was alarming, and it was really, really bad. There were no medicines. And I remember there were really harrowing images of patients with HIV and AIDS, and there was a lot of stigma. And growing up in that time, I really thought that something that I wanted to do when I grew up was find a vaccine for HIV. And interestingly, this hasn't happened yet, but the search for the vaccine of HIV has really opened a lot of new science and has really led to some of the discoveries that, for example, have also led to the vaccines that we have now against COVID-19. And a lot of the research that developed from HIV research has really propelled the biomedical sciences in a really, really fast way. So after high school, I really wasn't sure what I wanted to study. I knew I wanted to study something that would allow me to kind of do science, kind of answer questions. I wanted to understand the world and the phenomena around us, not only biological diseases and biological mechanisms, but everything. I was very curious. So I obtained a scholarship from the German government that allowed me to go to Munich, the Technical University of Munich. And I decided to study engineering physics, partly because I wasn't sure what I wanted to study, but also because engineering physics I thought would be a good basis for any kind of science and any kind of research. This is the Technical University of Munich. This is where I spent a lot of the time. There's a slide that goes from the 4th floor to the 1st floor, and everyone uses it, even professors. It's shaped like a parabol, so it's pretty cool. I'd say going to undergrad at first was hard, and I know a lot of you guys are in the transition to go from high school to college, and I'd say that first year was really hard for me, and I didn't pass a lot of courses. Moving from Costa Rica to a completely different country was a big change for me. I had never seen snow or winter, so I really did really poorly when I started, and I didn't pass courses, but And a lot of people told me maybe you shouldn't study physics since you're doing so badly. But I think I was really stubborn, and I knew that I could do it if I wanted. I just had to work harder. So one thing that I think I learned during this time is that you don't want to let those early failures define the rest of your studies or your career. It's something that happens a lot when you start. You might fail and you might think, okay, maybe this isn't what I should be doing. But sometimes you just have to push forward, and you can demonstrate to yourself that you can do it. So during my time as an undergrad, one of the things that I realized was that I didn't really enjoy classes much. I didn't enjoy the theory. I really wanted to understand how we can apply the concepts that we were studying in class to specific projects, to specific science, to building new knowledge. So I joined a lab that was working in DNA origami. And I don't know if any of you are familiar with DNA origami, but it's a really cool field where you use DNA not as genetic material, but you use it to build things. This is a pretty famous Smiley that was completely built out of DNA. So this is at the nanoscale. It's really, really tiny, but it's really exciting that we can use DNA to build something so small and so structured. And the way this works is we have a single-stranded DNA loop, and we can start adding pieces of DNA that are complementary to this loop of single-stranded DNA. We call this staple, but this is really just a piece of DNA that has complementarity to different parts of this DNA. And if you put a lot of these little staples together, you can create, for example, a rectangle here. But you can go beyond a rectangle. You can construct boxes, you can construct smileys, you can construct all kinds of different shapes, curvature. Here we have a robot, for example, that was constructed completely out of DNA. And it was a fascinating field for me and a fascinating first step into science and research as an undergrad. I'd say if any of you are interested in science and research, starting as soon as possible when you, as a freshman or as a junior, or as early as possible to do research is a really good way for you to set you up towards a career in science. All right, and if anyone has questions here before I move to the next step, I'm happy to take any.
Enrique, do you want to unmute?
Sure, sure. I was also wondering because I saw some pictures of like a box and a tetrahedron. Is it possible to use these staples to actually make like 3D structures?
Mm-hmm. You can definitely
Wow.
create 3D structures. This robot is a 3D structure, even if it doesn't look like. But you can see this model is actually a 3D structure.
Oh, that's so cool. And
It is.
if you don't also mind me asking, how rigid are these structures?
So what's really cool is you can actually control the rigidity of the structures by the amount of staples that you're adding to it, and also the curvature that you add and the type of the way you create the structure. It really allows for you to decide this design principles.
Ah, so cool. Thank you so much.
You're welcome. Any more questions? All right. So during my time working in physics and in DNA origami, I think there was still a part of me that really wanted to kind of go back to that HIV research. I was still thinking about the vaccine and how important it was and how we still had no vaccine for HIV. So I think as a junior or maybe as a senior, I applied to a bunch of different programs, both in the UK as well as in the US. I think I applied to about 30 different labs. Some of them were doing HIV vaccine research. And in the majority of those labs, the principal investigators said no, that my background in physics really didn't fit the research that they were doing. But one single lab out of those 30 in the UK at the University of Cambridge said that they would take me. It was a very young professor. He said if I could find my own funding, I could come. So I applied to a bunch of different funding sources, and I luckily got some funding from the European Commission that allowed me to go from Munich to the University of Cambridge for a year. And here, I really focus on epigenetics, or this was the first introduction for me of epigenetics. And I learned about DNA folding and organization, but from a different perspective, way more biological. And in this field, I learned that in each cell of the human body, we have over 2 meters of DNA. And this DNA is packaged in a very specific manner in the nucleus that has a diameter that's 300,000 times smaller than the actual length of the DNA. So it's pretty fascinating that something so long can fit in such a tiny space and that it can be functional. So during my time in the UK, I learned that DNA is packaged in a very specific manner. It uses these proteins called histones. DNA is negatively charged. These histones are positively charged. So this allows for DNA to wrap around these positively charged histones to allow for compaction of DNA in an organized manner. And what's really cool about these histones is that they have these tails that come out of the core. And these tails can be modified with different chemical modifications that really can control the charge of the charge of the histones. So you can add here different modifications that may make this less positive, and this would allow for DNA to be more open in some parts, or you can make it even more positive and make it even more compact in some other parts of your genome. And this is really important for different functions that I'll also talk about later on. But after my time in the UK, this really sparked in me an interest to go back more into biology and to think about genes and DNA. So I think in 2013, I applied to a bunch of programs, both in the UK as well as in the US for my PhD. And I only got into one out of like 20 programs I applied to at the University of Pennsylvania. Oh, and before I move on, if there's any questions, I'm happy to take them.
Yes, Vivian, go ahead.
Oh, so I should have asked this earlier, but what is the practical use of DNA origami in the forms of self-assembly?
That's a great question. So I would say most of the applications that people have been thinking about with DNA origami is measurements. You can create very tiny calipers, for example, that can measure distances. You can also create molecular rulers. So you can create a DNA origami of a specific size, and then you can put that into a cell, and that can allow you to know like how large different things within that cell are without disrupting the cell. Another possibility is, for example, the boxes that I showed you in the image, they can be loaded with drugs. So you could do delivery of drugs to specific cells using those DNA origami boxes. There are a lot of challenges when you use DNA as a vehicle, because the body doesn't really want or doesn't accept DNA that's not its own. So it will mount an immune response when it sees DNA origami structures, and it will digest those DNA origami structures. So researchers have developed different methods to kind of hide the DNA origami structures from the immune system. But there's still a lot to do.
And Fatima, if you want to go.
Okay, hi, I am also from Costa Rica. And what I wanted to ask you is whether you left for Germany on a scholarship, or if you did right after high school. And if so, how did you get one? I mean.
Yes, I left right after high school. I had the opportunity to go to a German high school in Costa Rica, which allowed me to learn German, and that allowed me to get this scholarship. But there are many scholarships from the German government. I think the main challenge is undergraduate courses are delivered in German. So you do have to know German to be able to go there. But once you're there, there's no tuition. So most of the scholarships cover living expenses. So it's much more affordable to go to Germany compared to other countries like the US or Canada or the UK. Even if you want to study medicine, it's free for foreigners if you can speak German. Another strategy is going during your master's. There's a lot of master's courses that are in English, and there's also a lot of scholarships.
Thank you.
You're welcome. All right. So as I mentioned, I applied to a lot of different programs and only got to one at the University of Pennsylvania. And I really was excited to go there because they have Epigenetics Institute, and I wanted to learn more about epigenetics after being introduced to this topic. And one of the questions that I always really been fascinated by, and I think it's one of the most fascinating questions in all of biology, is how from a single genome we can get over 200 different cell types, how a single fertilized egg gives rise to so many different types of cells, epithelial cells, neural cells, immune cells, cardiac cells, bone cells, a whole organism. It's really interesting because all of these cells in the core have the same DNA. So you can imagine that they have the instructions to make any of these cells. Why do they develop a specific identity, and how do they maintain that identity? So that was the question that I was really focused on during my PhD. How at the molecular level with DNA organization, what leads to identity, and how is this identity maintained? And this is an important question because, for example, in cancer, cells that aren't supposed to have properties like being able to go metastasize and go to different parts of the body, those cells shouldn't be able to do that based on their identity. So there's a change in identity. And a lot of those questions were things that I was interested in understanding. And what's, I think, at the core of this is what genes are expressed in each cell and which genes are not. And what's really important, I think, is that the genome, only 2 to 3% of the genome encodes proteins. So the vast majority of our DNA doesn't encode any genes. And for a long time, a lot of people called the rest of the genome junk DNA because they didn't understand what it was doing. But in the past 20 years, we've realized that about 80% of this non-coding DNA really plays an important role in regulating gene expression and regulating the expression of this 2 to 3% of the genes. And this is really crucial in terms of cell identity. So during my PhD, I focused on work that was studying enhancers. These are non-coding regions of DNA that regulate cell type specific expression and maintain cell identity. This sounds complicated, but in simple terms, what we observe, for example, is that there's a gene that's expressed, for example, in the brain as well as in the limbs. And the current model is that there are enhancers or non-coding regions of DNA that play an important role in folding towards where this gene starts and leading to activation of those specific genes. And this happens in a tissue-specific manner. So this only happens in the brain because there's specific proteins, transcription factors that can bind to these sites and activate the transcription of that gene. And then in the limbs, a different protein binds to a different enhancer to activate expression of that gene. So during my PhD, I focused on one specific transcription factor, one of these proteins that binds to those enhancers called P63. You can see that if you get rid of P63 in mice develop, they don't develop properly, they don't have limbs, their skin doesn't develop properly, they don't have eyelids. And then in humans, what we've seen is that substitution mutations in P63 can lead to very severe phenotypes where patients have cranial malformations as well as malformations in their limbs. And although this is a very, very rare disease, what's really common and what we observe in these patients is clefting of the lip and the palate. And that's one of the most common inborn defects in the world. 1 in 700 newborns is affected by cleft lip palate, which is an opening of the roof or the palate and an opening of the lip. So during my PhD, I really focused on studying this phenomenon. But also during my PhD, I realized that science is really important beyond bench science. And I really wanted to understand how we could use science for more. So I founded the Penn Science Diplomacy Group during my PhD to study how science fosters collaboration that crosses borders. For example, during the Cold War, science was one of the only channels that allowed for communication between the Soviet Union and the US. And it continues to be one of the easiest ways to communicate across countries that are in conflict. And then another thing that I worked on was a podcast called Caminos en Ciencia. It's entirely in Spanish. So all of you guys, Spanish speakers out there, you should check it out. It's a podcast where we interview scientists from all over Latin America. And what we're trying to aim here is to bring role models to everyone in our region. The same way that Franklin Chan was really important for me, we want to be able to provide those examples for people in our region. So if you speak Spanish, please check it out. If not, we also have summaries in English in our website. And then I really wanted to mention that during my time and throughout these different activities, I learned that it's really important to increase trust in science. And this is something that we observe right now as well with the vaccine. There's a lot of distrust towards science. And a lot of people don't want to get the vaccine because they think it happened too quickly, for example, or they think that it might not be safe. So I think as you think about your careers and if you're moving into science, it's really important to think about how to maintain and to increase trust in science. And 3 ways to do it is improving science communication, increasing diversity in science. It's important for communities to be represented in science so they can explain the unique concerns that those communities have to other scientists and also to the communities themselves. And then I think it's really important to be able to allow and accept uncertainty, which is something that people think comes naturally to scientists, but I would say there are a lot of incentives to not do this. And it's important to keep this in mind as you move into your careers. All right, before I move into CRISPR, I wanted to see if you have any questions.
Bonnie, do you want to go?
Yeah, sure. So I just wanted to clarify because I remember in like my biology class, my teacher had mentioned that it seems like only a small % of our DNA is functional because we consider that it is only read in one direction. But then you can also read it backwards per se. Is that like, do you have any like information on that?
I would say that it can be read in both directions, yes. And there are genes that are sometimes on top of each other because one is read in one direction and the other is read in the other direction.
Okay.
Yes.
Yeah, and another quick question I had is that in terms of the slide that you have up here, one important part of like science communication is also communication between scientists of different disciplines. And I know that sometimes one group of scientists are they don't see the importance of another field as much. So how do you suggest that we motivate these scientists to understand and embrace the importance of other fields?
I think that's a great question. I think a lot of science communication, as you mentioned, is focused on going to conferences where you talk to peers that are working in the same field. And there isn't an understanding of why other fields are equally important. I think one really important thing to remember is that most of the science that we do is paid by taxpayers, so the public. So there really should be an imperative for all scientists to communicate their science, not only to other scientists, but to the public in general. And I think what we're lacking is training in science communication. This isn't a big part of graduate programs. This isn't a big part of undergraduate programs in science. And I think it's really imperative that we change that and that we include this as a basic training for scientists.
Great, thank you so much.
Enrique?
Yeah, I'm gonna need to remember my question. How do you discover specifically which genes control like the expression and manifestation of certain characteristics? And how would you like pinpoint them and identify them to other researchers?
That's a great question. I'd say a lot of the genes that we've realized play important roles, for example, for identity, we've been able to identify because of disease. We see that there's a malformation in a human or an animal, and then we go searching, we go sequencing, for example, the functional parts or the exonic or the gene parts of the genome to see if we can identify mutations that might explain that. And then we can go back and introduce those mutations in a mouse model, for example, to see if we can recapitulate the same phenotype. And through processes like that, that take several years or used to take a long time, we're able to kind of understand that phenotype genotype connection. And then now with CRISPR, this has been accelerated and we can do this in a really, really quick manner right now. Instead of taking several months to make a mouse model, we can do it under a month now. So this really it's a game changer and allows us to ask this kind of questions. Exactly.
So cool. Thank you again.
You're welcome. All right. So on to CRISPR. So I think at the end of my PhD, I had been working on DNA across my entire career, from DNA origami to epigenetics to these questions about cell identity. And I was aware that DNA controls a lot of different traits, from whether we have cheek dimples or aversion to cilantro, clefting of the chin, dandruff, even things like fear of public speaking and finger length ratio or fear of heights. People have been able to identify different sequences in our DNA that might explain some of these characteristics. And then these are mostly, we've also identified mutations that can lead to disease like cystic fibrosis, protein deficiencies, hyperinsulinism. There are many different identified mutations that can lead to disease and can be inherited. And I think at the end of my PhD, I really wanted to move beyond being able to study and characterize DNA as it is, and be able to actually manipulate DNA and change its sequence, which brought me to CRISPR, this new technology that really allows us to change DNA. So I moved again from Philadelphia to the University of California, Berkeley, to the lab of Dr. Jennifer Doudna. She's here on this side. This is me holding Cas9. And I'm really excited to tell you a little bit more about CRISPR-Cas9. This technology was only, or the technology of Cas9 as a genome editor was only identified in 2012, but it's come a long way since. So CRISPR comes from its name, clustered regularly interspaced short palindromic repeats. And you can see already why we never use this. We always just say CRISPR. And I have a video here that shows you where CRISPR comes from. And I think what's really interesting is, especially in the time where we are right now with this virus, is that CRISPR originally is a mechanism of how bacteria can protect themselves from viruses. So bacteria, like humans, can be infected by viruses. And they have evolved different mechanisms to kind of defend themselves. In humans, we have the immune system. In a lot of bacteria, we have CRISPR. So here we have bacteria, which are these brown tubes, and we have all these viruses of different colors that are coming to attack this bacteria. We have this pink virus that's injecting its DNA into this bacteria. And if we have a bacteria like the one in this video that has a CRISPR system, it can cut out a little piece of this DNA from this virus and incorporate it into its own genome. And it incorporates it into a specific site in its own genome called CRISPR. This is where CRISPR comes from. We can see that there's a lot of pieces of other viruses that have infected this bacteria prior to this pink one. But really this CRISPR site acts a little bit as a library or a memory of all the different infections that have affected this bacteria in the past. So the next time that this bacteria experiences an infection, it can make a copy of this DNA region. This is called CRISPR RNA. It can cut it into pieces, which correspond each to a different virus. And here we have a different RNA that comes to play called a tracer RNA. This tracer RNA allows this to be loaded into a protein, Cas9, which acts as a machine that goes all around the bacteria, looking for the sequence that has infected it in the past. And if it identifies the sequence or this virus, it can bind to it and produce a double-stranded break. And this is the way that bacteria really elegantly can protect themselves from viruses. And it's what we exploit now for genome engineering. Nope. Okay, and here I have an analogy to maybe make this a little bit easier to understand. If we think about the human genome and we were to paste the entire human genome with its 3 billion bases on Word, we would cover over 1 million pages. So picture 1 million pages of a Word document. And then CRISPR has the ability to go anywhere within that 1 million pages and identify a sequence of 20 letters almost anywhere in that 1 million pages and produce a double-stranded cut right there. So this is really fascinating, and this ability really allows you to do genome editing, because the first step in editing, if you're editing like a video, if you're editing a book, the first step you want to do is be able to cut, and then you can paste new things, you can replace things. So this really is the start of editing, and it's really an exciting tool that we now are able to use. And this fascinating ability has really led to this being awarded the Nobel Prize in Chemistry in 2020 to my boss, Dr. Jennifer Doudna, as well as to Dr. Emmanuel Charpentier for the development of a new method for genome editing. And there were two key discoveries that allowed CRISPR to become even more versatile. The first one was, as I mentioned, there's two pieces of RNA, the tracer RNA and the CRISPR RNA. The first discovery was that you could fuse these two to create a single guide RNA. And this really facilitates the use of CRISPR. But the most important thing that was discovered is that you can really change the sequence of this CRISPR RNA. This is a sequence of 20 letters. You can basically change it to any sequence, which allows you to target or to create a cut almost anywhere within the genome. And here we have a quick video that shows us CRISPR-Cas9 in action. We have DNA here, we have the protein here. And we can see that within seconds, CRISPR-Cas9 can create a double-stranded break. You see it here. It releases the cleaved DNA. Okay, and here's a video of how we can then use CRISPR-Cas9 and this ability to cut at a very specific site to do genome editing. Here we're looking at a human nucleus, and we're slowly
scientists have long imagined
moving into the nucleus of that cell. We can see DNA is packaged around all those nucleosomes, and we have different CRISPR proteins here looking for the sequence that we program it to look for. And if it finds the sequence that we've programmed it for, it can bind to that sequence, open the DNA, and create a double-stranded break at that specific site. And the other thing that we can see in this video is that we can then use the same DNA machinery, repair machinery in the cell to insert a new piece of DNA into that specific site. And this is really exciting. For example, if we think about genes that could predispose a person to having higher risk to developing cancer, like BRCA1 mutations, we could not only remove those genes, but we could replace them for one that's not mutated. And what's also really revolutionary and what makes CRISPR such an exciting tool is that if an organism has DNA, we're able to edit it. Here we have different organisms that are really common to labs, for example, mice, zebrafish, Drosophila, C. elegans, yeast. We've been able to use CRISPR to edit these organisms, but we've also been able to edit over 280 different organisms already using CRISPR. And this really has led to CRISPR being a toolbox with really diverse applications. And this opens completely new possibilities. For example, In drug development, as Brian mentioned, we can use, or CRISPR has been used, for example, in the most current vaccines. We also have animal models that have been developed using CRISPR. Instead of having to wait months and months to get mouse models of human diseases, we can now do them in less than a month. We are able to study genetic variation, and really some fields where this CRISPR technologies have been really important is, for example, in agriculture. We can now create maize or corn that might be resistant to climate change. We can also create coral that might resist ocean acidification. So CRISPR really brings new possibilities in terms of what we can change and what solutions we can now propose for some of the big challenges that we have right now. This is from 2018 and it describes a CRISPR toolkit, and it's already a little bit outdated. We have more tools already, but this has the basic tools that we have for working using CRISPR. We have Cas9 and Cas12, which is a similar protein as Cas9. We have also new proteins called base editors that allow us to, instead of producing a double-stranded cut, just switching one specific letter in the DNA for another. We also can use Cas9 for imaging DNA, for example. Another way we can use CRISPR is to modulate gene expression. We can use a Cas9 that doesn't cut to bind to specific sites. And if we've used activators or repressors to that specific Cas9, we can activate or repress gene expression. We also have the ability now to directly target RNA instead of DNA using, for example, Cas13, which is a CRISPR system that recognizes RNA instead of DNA. I'm happy to take any questions here as well, if you have any.
Vivian, go ahead.
I'm just wondering like what technology was used to film that Cas9 making the double-stranded break.
Oh, I'm not really sure which technology was used, but I can certainly figure that out.
Andre.
Good evening. Can you please discuss quickly the ethics of editing developing humans? Things such as preventing birth defects, getting rid of genetic fears, but also something that could be commercialized, like improving aesthetics or physical ability.
I will get to the ethics later on.
Okay, thank you.
No problem.
Lina.
This is a bit of a similar question, but as CRISPR is getting more widely used, do you think there would be a negative implications to biodiversity on Earth?
That's also a question that I will discuss a little bit later, but it is a possibility that CRISPR could be used to change ecosystems, and that would definitely have an effect in biodiversity.
Jody.
Let's jump to Leonardo.
Okay, Leonardo, go ahead.
Okay, good evening. What are your thoughts on applications like the gene surgery you showed, along with the rise of personalized medicine alternatives?
I'll also discuss some of the therapeutic uses of CRISPR in a little bit. So we can also go back to those questions afterwards.
Okay, thank you.
Most of the rest of the questions are about the ethics. So do you want to put a pin in them and then go back to them kind of later?
Okay.
Let's do Adams then, because I think that's a little different.
Okay, Adam, yes.
So do you think CRISPR could potentially be used for de-extinction purposes? Because I know it would take a lot of editing, but like if the technology was somehow like made more efficient, do you think that could be it?
That's a great question. And I think one of the poster child of that is mammoth. there is a researcher called George Church at Harvard who's trying to bring back the holy mammoth. And I do think that CRISPR technologies might advance to the point where that might be possible. The question is whether that's an important question that we should be working towards as well. All right, I'll move on, but then we can go back to some of the ethical questions as well as the applications. As I mentioned, CRISPR has, well, it only was developed as a gene editing tool in 2012. And what's really exciting is that it has already been used to treat human patients. The first example of this is sickle cell. This is a disease where red blood cells have a sickle shape instead of their normal round shape. And this leads to the sickle cells really becoming obstructed in a lot of different places in the body, and patients having a lot of severe pain. Patients really require a lot of transfusions throughout the year to be able to cope with some of the side effects of this disease. And this is really only due to a specific single point mutation in these cells. So in 2019, the first patient in the US to be treated with CRISPR is Victoria Gray. She received a treatment for her sickle cell disease. And I think the last update that I heard from her was that she's doing really well. She hasn't needed any blood transfusions and other care since she got this treatment. So it has really changed her life, and it has shown the power of this technology. Another example, one of the things that they mentioned here is that the way you treat these patients is by removing the cells, editing them outside of the body, and then putting them back into the patient. But we also saw editing in vivo, so directly into the body, to treat genetic disease. This is a specific kind of blindness that's due to a point mutation. And doctors started the first trial last year to treat these patients using CRISPR-Cas. The results of this trial have not been published yet, so we don't know yet, but it's a really exciting time for these strategies. And then we have more strategies that are being developed, for example, in progeria. This is a disease that has no treatment. There's no current treatment, and it leads to early aging of tissues and patients. And what we've observed is using one of these base editors that does not create a double-stranded cut, but switches one specific base, they've been able to at least rescue this phenotype in mice. So we're hoping that this can then move towards therapies for patients in the near future. And then, as I mentioned as well, one of the places where where the use of CRISPR is really clear is in agriculture. The Food and Agriculture Organization of the United Nations expects that the global population will reach about 9.8 billion by 2050. And this will require a rise by 70% of food production to be able to feed everyone. And then this projected increase will really lead to rising energy prices, growing depletion of underground aquifers, loss of farmland, increased drought and flooding, and climate change. So there's really a need for solutions in terms of food and agriculture. And CRISPR might provide some of the solutions. I have an example here, which is Cassava or yuca, as we call it in Spanish. This is a threatened staple crop in the tropics. Cassava is to African peasant farmers as rice is to Asian farmers. It feeds nearly 1 billion people annually, about 10% of the world population, and it's difficult to breed because it has unpredictable flowering. So you might wonder why edit cassava. Cassava, similarly to people or bacteria, can be affected by disease. Here we have cassava brown streak that makes cassava inedible. So we can use genome editing to reduce susceptibility towards these diseases and maybe eliminate susceptibility to those diseases. The other important thing is that cassava also has cyanogens, which in large quantities might be toxic. So we can use genome editing to reduce cyanogens in cassava to create safer cassava for consumption. And for those of you who are not familiar with cassava, it's what it's used to make the bobas in bubble tea. So yeah, now we move to the ethics. And this is really an important question. Now we can edit DNA, so what should we edit? And there's important questions here about germ cells. These edits can be inherited from one generation to the next. We have also questions about gene-modified organisms. What agricultural products should we change? What could happen to biodiversity, as one of you guys mentioned? What could happen if we ended up with only monocultures of specific organisms? And then one intriguing possibility that has also, it's also allowed by CRISPR is gene drives. This is a way to eliminate an entire population from an ecosystem. And this is being developed as a possible way to eliminate, for example, the mosquitoes that carry malaria. You can use gene drives to kind of eliminate them from a population and then that way eliminate the risk of malaria. But the question is, should we use this and what consequences could this have for the environment and for humanity? In terms of human germline editing, there's many questions here about the ethics of using CRISPR for human germline editing. And here I think it's really important to discuss the difference between somatic and germ cells. Most of the all of the treatments that I mentioned today that are using CRISPR in humans to date is in somatic cells. These are non-heritable changes. It's one individual that's affected. But if we were to edit germ cells, this would be heritable. This is not only the individual, but also the offspring that's affected. And these changes can then enter the gene pool. There's a lot of ethical questions about the germline. There's technical risks, there's philosophical risks, there's social risks. In terms of technical risks, there's still, even though technology is really exciting and it really allows for a lot of different changes. One of the things, for example, that we do not have 100% control of is off-target effects. Sometimes when we're trying to create a cut somewhere, we might, by accident, create cuts in other parts of the cell. And this could lead to off-target effects that might lead to cancer, for example. Other technical risks are mosaicism. Sometimes if you were to introduce Cas9 into an egg or a sperm or a zygote, you can have some cells that are edited, but not all of them, which can lead to a person having or becoming a mosaic where some of its cells are edited while others are not. There's also philosophical risks. There's religious reasons. There's also a philosophical question, which is how we're treating someone who cannot consent to treatment, and should we be allowed to do that? There's also, as someone mentioned, a clear distinction right now between treatment versus enhancement. A lot of current researchers are focused on this part right now. We want CRISPR to be used to treat diseases that are currently untreatable. There's no cure. Diseases that are really truly devastating and where there's no other solution right now. However, once you allow some of these treatments, this might create a gray zone where people might start thinking about, well, is it possible to then enhance the genetic material for a specific person, create a person that's stronger, for example. And this really opens up a lot of questions about what's treatment, what's enhancement, what happens when a specific characteristic that we consider enhancement right now becomes mainstream, is not having that characteristic then a disease. So this really opens a lot of questions that are important to address. And then there's also social risks. A lot of these technologies are expensive and might not or will not be available for the vast majority of the population, at least not initially. And this really creates ethical questions and concerns where only people who can afford it might be able to access these technologies. And then the inequality that's already huge would be not only in terms of money, but it would be encoded within the genome. And that's, I think, a really scary thought. So a lot of these questions have been addressed in the Human Genome Editing, Science, Ethics, and Governance report that was developed in 2017, where I think a moratorium was set for no human embryo editing, and that's currently the stance. Human embryo editing is not allowed right now for many of the reasons that I mentioned before. However, in 2018, a Chinese researcher edited human embryos for the first time. And those embryos further developed and were born. So this really has changed the focus to something that's possible, to a reality now where it's not only is it possible to edit human embryos, what would that mean? But the question is now, like, this has now happened. What should we do now? How should we regulate these technologies? How can we stop this from happening again? And how can we move away from designer babies, where people might want to choose their offspring's eye color, their offspring's height, all these different questions that are becoming, it's important to discuss now before the technology advances more in terms of embryo editing. And yeah, as I mentioned, there is really an urgent need to bring down the costs of therapies. And this is also evident with the current vaccination efforts, where at least if you live in the US, you have access to the vaccines, but in many other countries, including my own country, Costa Rica, a vast majority of the population does not have access to the vaccine. And if we can't deliver something that's so important to all of us, what's going to happen with technologies like genome editing that are even more complicated and more expensive? But I think I wanted to end in a lighter note before we move to questions. I wanted to discuss that really, despite the questions that arise with CRISPR, the possibilities that it has afforded are really endless. In basic research, in medicine, in agriculture. For example, in basic research, it has really allowed us to ask and answer questions that we didn't have tools to before. Here's an example: butterflies and their patterns. We really didn't understand how butterflies chose their patterning. And now using CRISPR, researchers have been able to answer some of these questions. Another question that I think is really out of the box and interesting where CRISPR has been used is why humans walk on 2 legs instead of 4. So people have introduced changes into different rodents to try to understand or try to mimic some of these processes. In medicine, as I mentioned, there's new therapies. There's a lot of challenges in medicine, like antibiotic resistance or new viruses, where CRISPR can play an important role. And then in agriculture, as I also mentioned, there's so many different possibilities here in terms of how CRISPR can be used. And then I also wanted to talk really briefly, as I mentioned, about the COVID-19 pandemic. Most of the applications that have been used or I've talked about today are from CRISPR-Cas9, which was identified and developed as a genome editing tool in 2012. But since then, we've identified and created many more genome editing CRISPR systems like Cas12s, Cas13, which I mentioned recognizes RNA. And then even just last year in our lab, in the Doudna lab, we identified Cas12J, a really small genome editing nuclease. One of the really interesting things is that all of these different CRISPR systems have different properties. And one that was really interesting is collateral cuts. This is, for example, a Cas13. This recognizes RNA, Once it identifies its target RNA, it becomes completely activated or in an active form, and it starts to cut any kind of RNA that's close to it. And this has now been used to create diagnostics systems. And I have an example here of how CRISPR, even though it hasn't been used directly to attack the SARS-CoV-2 virus, we've been able to use it to detect it. We have, for example, patient samples like blood, urine, cells, and we're looking for DNA or RNA. In the case of the COVID-19 virus, SARS-CoV-2, we're looking for RNA. We can use Cas13 that detects the specific RNA from the virus. And once it does, we can add here a probe that has a quencher and a fluorescent probe. This fluorescent probe does not fluoresce because this molecule here is quenching its fluorescence. But once this Cas13 becomes activated, this collateral phenomenon that I talked about will cut this in two, and this will lead to the release of the fluorophore and signal emission that allows us to detect SARS-CoV-2 in the patient sample. And what's really cool is that this can be used for not only detecting infections, but also detecting cancer or different SNPs, single nucleotide polymorphisms, or changes in the sequences of DNA that can lead to diseases. And then really finally, I wanted to, in 3 slides, talk about some of my work. My research, as I mentioned, has always revolved around DNA, from DNA origami to epigenetics to genome editing. Right now, the question that I'm trying to answer is how do we efficiently and precisely insert large segments of DNA at targeted sites? CRISPR is really good at cutting, that cutting function, but it's not as good at the pasting function and putting new things into the genome or replacing genes. So really my work is focused in trying to understand how can we develop processes that improve that part of the technology to do, for example, repairing deletions, replacing exons, or gene replacements, engineering cell-cell communication, creating endogenous tags and tools to answer basic biological questions, and also improving therapies like CAR-T immunotherapy. All right, and if you have questions about our work and our science, you can also visit our website, innovativegenomics.org, or visit us on social media.